The overall goal of this model is to simulate apnea-caused hypoxia and hypercapnia in a human model with a focus on intermittent apnea, which occurs during sleep apnea and emergencies such as airway obstruction.
This model may help to understand physiological mechanisms to avoid hypoxic damage to the brain despite prolonged apnea. The main advantage of this dynamic model is that we could simulate clinically relevant hypoxia in humans.
Apnea-dependent hypoxia often decreases heart rate. Hypoxia caused by lowered oxygen in the inspired air increases heart rate. Thus, different models of hypoxia induce opposite effects in circulation.
Our reproducible model enables us to investigate different technical devices. This gives us the opportunity to focus on different physiological aspects of apnea without changing the setup or model.
Prior to attaching electrodes to a subject, clean the attachment points using 70% alcohol. Now, place an NIRS electrode over the frontopolar 2 locus on the right side of the forehead above the eyebrow and to the right of the mid-sagittal sulcus. Use a second electrode to measure the peripheral tissue oxygenation. Place it on the inside of the lower arm. Avoid placing this electrode above a venous plexus.